We're still in chapter 23, and this is going to be over the liver, gallbladder, and pancreas. All three are accessory organs associated with the small intestine, and the liver's digestive function is to produce bile, and bile is a fat emulsifier. The gallbladder's major function is to store bile. A lot of people get that confused and think that the gallbladder is the organ that actually produces the bile, when in fact it just plays a role in the storage of bile. And then the pancreas supplies most of the enzymes needed to digest chyme, as well as bicarbonate to neutralize stomach acid. The liver is the largest gland in the body. It weighs about 3 pounds, and it consists of 4 primary lobes. And here is the liver. I'm not going to ask you about any anatomy of it on the liver as far as like looking at a diagram on the exam. That's something that you're going to do in lab. But I did want to show you an image of the liver, and then this would be the underside of the liver. The lesser omentum anchors the liver to the stomach, and there's something called a porta hepatis, and this is where the hepatic artery and vein enter the liver. The common hepatic duct leaves the liver. The cystic duct connects to the gallbladder, and the bile duct is formed by the union of the common hepatic and cystic ducts. And so that's what we see here. Cursor. This is the cystic duct. This is the common hepatic duct, and this is the bile duct. The microscopic anatomy of the liver consists of liver lobules, and these are hexagonal structural and functional units, and they're composed of plates of hepatocytes, which are liver cells. And these are going to filter and process nutrient-rich blood. And then in the middle of the liver lobule, we would find a central vein. And so these are the lobules. These are the hexagonal structures, and this is going to be the central vein. And we see a blown up image of this, and we can again see this is a central vein, and this is the lobule. We're also going to find liver sinusoids, and these are leaky capillaries between hepatic plates, and stellate macrophages or hepatic macrophages. And liver sinusoids are going to remove debris and old red blood cells. So again, this is a liver lobule, and this is a central vein. These are going to be the sinusoids, and then in here we find the macrophages. Hepatocytes have an increased rough and smooth endoplasmic reticulum. They, in addition, have an increase in the amount of Golgi apparatus, peroxisomes, and mitochondria. And the hepatocyte functions include producing almost a liter of bile a day, so about 900 mils of bile per day, processing blood-borne nutrients. So we've talked about how the liver stores our glucose as glycogen. We talked about that in AMP1. And then it stores fat-soluble vitamins and performs detoxification. For example, converting ammonia, which is very toxic, to a less toxic urea. Bile is a yellow-green alkaline solution that contains bile salts, and bile salts are a cholesterol derivative that function in fat emulsification and absorption. Bilirubin, and this is a pigment that's formed from heme, which is from the breakdown of red blood cells. We talked about this in chapter 17. And the bacteria in the intestine break that down to stercobilin, and that's what gives the brown color of feces. And then we would also find cholesterol, triglycerides, phospholipids, and electrolytes. So now we move on to the gallbladder, and this is really all I have to say about the gallbladder as far as the organ itself. It's a thin-walled muscular sac. It's on the ventral surface of the liver, and it functions to store and concentrate bile by absorbing water and ions. And then we move on to the pancreas. The pancreas has both exocrine and endocrine functions. The exocrine function is to produce pancreatic juice. So there are these acini, and these are clusters of secretory cells that produce zymogen granules containing proenzymes. And then there are ducts that secrete to the duodenum via the main pancreatic duct. And smaller duct cells produce water and bicarbonate. And then the endocrine function we talked about in chapter 16, that's the secretion of insulin and glucagon by pancreatic islet cells. So this is an acinar cell right here. And then we would see the zymogen granules. These would be the duct cells. And then you can see the small ducts here and here. There we go. Again, acinar cell, and then the pancreatic duct. So pancreatic juice. So now we're going to talk about the composition of pancreatic juice. And about 1,200 to 1,500 mils per day is produced, and it contains a watery alkaline solution about pH of 8, and this is necessary to neutralize the acidic chyme that's coming from the stomach. It also contains electrolytes, primarily bicarbonate, and digestive enzymes such as the proteases, amylase, lipase, and nuclease. And proteases are going to be for proteins, amylase would be for carbohydrates, lipases are for lipids, and nucleases are for nucleic acids. Proteases are secreted in an inactive form. They're activated after they reach the duodenum. And we're going to talk about enteropeptidase. This is an enzyme bound excuse me, to the plasma membrane of the duodenal epithelial cells, and it's going to activate pancreatic protease trypsinogen to trypsin. Once trypsin is activated, it can then activate more trypsinogen, and it can also convert procarboxypeptidase to the active carboxypeptidase, and chymotrypsinogen to the active chymotrypsin. And so we can see that we have the inactive forms being secreted, and then they're going to be converted to the active forms. The bile duct and pancreatic duct unite in the wall of the duodenum. They fuse together in a bulb-like structure called the hepatopancreatic ampulla. The ampulla opens into the duodenum via a volcano-shaped major duodenal papilla. The hepatopancreatic sphincter controls the entry of bile and pancreatic juice into the duodenum. And then there's also an accessory pancreatic duct, and this is a smaller duct that empties directly into the duodenum. And we are looking for, let's see, yes, cursor. Okay, so this is the hepatopancreatic ampulla and sphincter here. And you can see this is where the two are joining. This is the bile duct, and this is from the pancreas. Regulation of bile and pancreatic secretions. They're both stimulated by neural and hormonal controls. Hormonal controls include cholecystokinin and secretin. And then bile secretion is going to be increased when enterohepatic circulation returns a large amount of bile salt, or secretin from the intestinal cells exposed to hydrochloric acid and fatty chyme stimulates the gallbladder to release bile. What this means is eating a fatty meal will stimulate the production or the release of bile, not the production necessarily, but the secretion of bile. The hepatopancreatic sphincter is closed unless digestion is active, and bile is stored in the gallbladder and released to the small intestine only with contraction. And here's your question. That's all I have for this talk.